In the previous video, I have discussed 25 questions from KSET 2021. I will be continuing the same question paper from 26th question. Proteins destined to be secreted move through the secretory pathway in which of the following order? So here they are talking mainly regarding the endomembrane system where a group of cell organelles work together to perform a particular function particular function. This one will be called as endomembrane system. Which cell organelles are involved in this endomembrane system? That is endoplasmic reticulum, Golgi complex, lysosome, and vesicles. So these are the main part of this endomembrane system. What actually happens in this endomembrane system? I will explain here. The rough endoplasmic reticulum, the rough ER, where the protein synthesis takes place. This produced proteins has to be transported to Golgi apparatus for the modification of this protein. So this protein will be transported via, these are the proteins, and this structure is called as vesicle, and this vesicle will go and reach the cis phase of the cis phase of the cis phase of the Golgi complex. This is a structure of a Golgi complex. Soon after reaching the cis phase of the Golgi complex, it will bind to that one so that proteins will get entered into this Golgi complex where the complete modification of the protein takes place in the Golgi complex. After modification of the protein, this protein will get eliminated in the form of secretory vesicles. These are called as secretory vesicles. So these secretory vesicles will go to different places in a cell. They may reach to the cell surface for the transport, or they may enter into the different parts in the cytoplasm to form a cell organelles, cell wall, etc. So this is the pattern how the particular function takes place in this endomembrane system. Here they have given option a rough endoplasmic reticulum, Golgi transport, vesicles. Golgi has been transported the vesicles. This Golgi cisternae, then secretory vesicle, then cell surface. So B is the right answer for this question. Then read out the next question. The unit of distance in linkage map is, that is centimorgan. C is the right answer. It is one of the repeated question. Then 28th question. It is also repeated question. The gene that is not involved in the body patterning in Drosophila. So not involved, that is SXL gene. This is not involved in the patterning of the Drosophila, where gap genes are there, segment polarity genes are involved, then and pair rule genes are also involved. Where in this Drosophila, the body patterning will be done through these different genes, where the particular maternal effect genes, segment polarity genes, and homeotic genes. These will be responsible in formation of the head, that is anterior part or posterior parts in the Drosophila. So this X, that is SXL gene is not involved. So C is the right answer. Then 29th, hemophilia is a sex-linked recessive trait in humans if a father and a son are both hemophilic but the mother is normal, her genotype must be. So here the father, he is hemophilic, whereas mother is normal, but but she may carry the gene, that too only one. Due to that she will be considered as a carrier. So carrier here, they are actually talking regarding the sex linked recessive trait. Here the gametes which we obtain, that is X and Y, and recessive H. So if the sperm containing Y fuses with the gamete produced by a male, then he will be what? A hemophilic. Hemophilic. So the mother will be having a this kind of genotype where it is one of the carrier in case of the mothers. 29B is the right answer. Then 30th question. Fluorescent recovery after photo bleaching, FRAP is a powerful technique for calculating. So this FRAP is one of the diffusion coefficient of our membrane lipids and proteins is the right answer. Then 31, at which cell cycle checkpoints is the cell cycle halted if the cell's DNA is damaged. Here, this is the diagram of a cell cycle. This one will be M phase, G1, and this one will be Z0. This one is S, and it is G2. Here at S phase, the replication of DNA takes place as it is a synthetic phase. Here, if the cell's DNA is damaged, we have a checkpoints. If a DNA is damaged here, then it will be halted between the S and G2 as we have a checkpoint here. So S and G2 is the right answer. Then next question. Which of the following occurs in meiosis but not in mitosis? So here they have given attachment of spindle fibers to kinetochore. It occurs in both. So it is not true. Pairing of homologous chromosome at the metaphase plate. So this one, it will be seen only in case of the meiosis. Meiosis. That too in case of the one. Meiosis, where the actual pairing of homologous chromosomes at the metaphase plate takes place. Replication of DNA prior to the start of cell division, it is not a correct explanation. Separation of sister chromatids at anaphase takes place in both. So B is the right answer. Then next question, 33. Next question. Which of the following techniques could be used to demonstrate protein binding to specific DNA sequence? So here they have given four different options: Western blot, Northern blot, Southern blot, and electrophoretic mobility shift assay. Here they are not at all talking regarding any protein or RNA, how this RNA protein get separated. They are not talking. They are actually speaking regarding the used to demonstrate protein binding to specific DNA sequence. How a particular protein will come and bind to this particular DNA sequence. This can be done through this electrophoretic mobility shift. This will tell regarding the how a particular protein can bind to a DNA sequence. Here Western blot is done for the separation of protein, Northern blot is done for the separation of RNA, Southern blot will be done for the separation of DNA. So 33 D is the right answer. Then 34th question: which eukaryotic cellular organelles are believed to have evolved from symbiotic bacteria? Symbiotic bacteria, that is a prokaryotes. A prokaryote. Here, in case of the some of the cell organelle, we come across a ribosomes, and also they have their own DNA. So, in case of mitochondria and chloroplast, we will be seeing the ribosome, which is, that is 70s ribosome. 70s ribosome will be seen in these two. Due to that, it will be said that it is evolved from the symbiotic bacteria. So C is the right answer. In case of prokaryotes, also we come across prokaryotes will have a 70s ribosome, whereas eukaryotes will be having 80s ribosome. Then 35th question: which cellular organelles are involved in the initiation of the intrinsic pathway of apoptosis? So this apoptosis, it is called as a programmed cell death. Cell death. If a cell is worn out or damaged or old, so these cells has to be removed from a particular organism. So this will be done through the special process, that is programmed cell death. So this programmed cell death is done by a special cellular organelle, that is mitochondria. This mitochondria will start doing this activity. So C is the right answer. These ER, lysosome, peroxisome, they are not a part of this apoptosis. Then 36th question: people who have an extra chromosome may have gotten it through, that is a disjunction or the non disjunction of the chromosome. Uh, if you consider a father and a mother, they will be having a 46 chromosome, where 44 autosomes will be seen and 2 sex chromosome will be observed. In the same way, in case of mother, 44 autosome plus the 2 sex chromosome. If you divide this one, you will get 2 gametes. 22 plus x, it is one gamete. Here 22 plus y, it is another gamete. So it is a normal pattern of the separation of the gametes during the meiosis. But in some of the individual, this separation of gamete will be affected. For example, I will be talking regarding the Klinefelter syndrome, where one gamete will be having the both sex chromosome. Another will be with zero sex chromosome. When it fuses with the sperm, it will form a 44 plus XXY. It is because of the non-disjunction. Non-disjunction means they doesn't undergo a distinct separation at the or during the meiosis. So B is the right answer. So here it is happened. This is happened because of the non-disjunction non-disjunction, and it leads to some of the disorders. So B is the right answer for this question. Then 37th question, embryonic stem cells are obtained from inner cell mass of the dash as a precursor. So inner cell mass will be seen in this a blastocyst. So outer trophoblast layer trophoblast layer and inner, so this one will be called as inner cell mass. This inner cell mass will form a future embryo. So this one will be seen in case of what, that is blastocyst. It is a typical structure of blastocyst where you will get a outer trophoblast. This trophoblast after tra- sorry after implantation it will go and bind to the uterine wall, whereas the inner cell mass it will get converted into embryo. So this inner cell mass will be having a totipotent character or a stem cell character. So D is the right answer. Then next one, 38th question, what is the natural function of type 2 restriction enzymes? So this type 2 restriction enzyme, where they will help in the protecting bacteria by cleaving the DNA of infected virus. So A is the right answer. Then HIV infected T cell carries the dash antigen on their surface. So that is in case of HIV infected T cell, where you will see antigens. On these antigen they will be having a surface that is CD4. So CD4, where we'll come across this a killer T cell. So these killer T cells will be having this surface antigens, that is CD4. CD4. So C is the right answer. Then 40th question, which of the following technique is considered as environmental friendly to clean up organic pollution and toxic waste? That is bioremediation. Bioremediation is the one of the best environmental friendly technique through which we can eradicate this organic pollution or the toxic waste. That is C is the right answer. Then read out the 41 question. Genes that are evolved by duplication but may not have identical function are called as. So these are called as a paralogous. If they are identical, then they may call it as homologous. Here, evolved by duplication may not have a identical function. That is a paralogous. D is the right answer. Then Warburg effect is characterized by. So this one will be called as Warburg effect, that is because of the increased glycolysis. So 42 is the right answer. Then 43, secondary metabolites are produced in which of the following stages? So secondary metabolites like drugs, flavonoids, antibodies, etc., they will be produced during which stages? That is during the phase. Stationary phase. This is lag, a log, and stationary phase. During this stationary phase, we'll come across the production of secondary metabolites by the bacteria in a colony. So C is the right answer. Then 44, a haploid sperm from one species and a haploid egg from another species may form a diploid interspecies hybrid. Meiosis in these plants generally fails but can lead to rare duplicated gametes called, that is, allopolyploid. Allopolyploid. So this diploid will get formed or the interspecific hybrid. After that, this meiosis it fails. If meiosis fails, then it will continuously multiply. That is, allopolyploid is the right answer. The next question, 45th. Epistasis is an example of or example for, that is, deviation of the Mendelian inheritance. C is the right answer. Then a head and tail region of the lipid in the cell membrane bilayer is. So this is a structure of lipid, that is phospholipid bilayer in a cell membrane. So these are the phospholipid bilayer, where in this phospholipid bilayer head head is the head is the hydrophilic part. Hydrophilic, that is water loving. Water loving. Whereas tail, they are hydrophobic. They have phobia of water, that is negative to water, or we can call it as non polar part of the phospholipid. This one will be called as polar head. So head is a hydrophilic, tail is a hydrophobic. This one is true. This one is true. So A is the right answer. Then 47th, according to Neo Darwinism, unit of evolution refers to, that is, population. B is the right answer. It is a direct question. Extreme phenotypes are favored over other phenotypes is called as. So where where extreme phenotypes are favored over other phenotypes. So this graph may shift according to the changes. So that is directional selection. That is extreme phenotypes akkadi valla ikkadi valla. So they may shift. So D is the right answer. That is directional. The population stabilizes on a particular non-extreme trait value is called as stabilizes. A population stabilizes on a particular non-extreme trait. That is, the graph will remain stabilized. So, that is stabilizing natural selection. This is called as stabilizing natural selection. So, C is the right answer. Then, next one. Which of the following not a parasite of human? So, it is a parasite. Sorry. So, parasite that is, it will cause a disease that is malaria. Trypanosoma brucei, it will cause a sleeping sickness. Sleeping sickness. Fasciola hepatica, so again it will cause a disease, some other diseases related to intestine. Then Lumbricus terrestris, it is a name of earthworm. So it is a friends of farmer. It is not an parasite. So D is the right answer. D is the right answer. Then 51. A specimen which have been referred by the author as similar to type specimen is known as, that is, referred by author as a similar to type specimen is called as, that is, isotype. That is isotype. The next one. In vertebrates, the combination of PAX6, SOX2, SOX3, and L-MAF transcription factors in the ectoderm of optic vesicle ensures the production of, that is, lens. B is the right answer. 53. Which of the following is not a biodiversity hotspot of India? Western Ghat, it is a hotspot. Himalayas, hotspot. Andaman Nicobar, it is a hotspot. The Chambal Range, it is not an a biodiversity hotspot. So D is the right answer. Then, what is the penetrance of polydactyly if 64 people have normal fingers and 16 have abnormal number of fingers? That is 0.25. So as we calculated here, 64 people is to 16, 16 only 16 ನಾಲ್ಕಲೆ 14. So that is 0.25. So A is the right answer. Then 55th question. Casual agents of bacterial blight of paddy is. So this bacterial blight of paddy is caused by Xanthomonas oryzae. Paddy means oryzae. So D, Xanthomonas, it is a scientific name for this blight disease. D is the right answer. Then 56th question. Which of the following is maternally contributed RNA in the early development of Drosophila embryo? So in this maternal effect genes, where we will come across bicoid, hunchback, So bicoid and hunchback, they play a major role in the formation of anterior parts of the body, that to head and thorax. So C is the right answer. 57th question. An affected female marries a normal male, and if half their daughters and sons are affected, the trait is follows. The trait follows. The trait follows. That is, affected female marries some normal male. If half their daughters and sons are affected, the trait follows. That is autosomal dominant. So D, B is the right answer. Then 58th one. Which one of the following enzyme cleaves RNA in an RNA DNA hybrid? So that is RNAH is the right answer. D is the right answer. Then 59th. Tata box or Pribno box are sites of. They are the sites of the promoters. C is the right answer. 60th question. Chemical responsible for Bhopal gas tragedy, that is methyl isocyanate. So that is A, gene and methyl isocyanate. So that is Bhopal gas tragedy, where the leakage of methyl isocyanate and gene. These are the two chemicals which got released during the tragedy. So A is the right answer. Then 61 question. Species found exclusively in ecotone are, that is edge species. Ecotone, here one kind of system, ecosystem, here another kind of system, where they will meet in between. This part will be called the meeting of two different systems or the ecosystem, that is called as ecotone. So this one will be called as, where we will come across this edge species. B is the right answer. Then we'll solve some more question in the next video.